Hi guys, so I'm in front of the Human Sciences building where I currently do research at. So let's go in and show you where it's all at. Now before I begin this lab tour, I invite you to check out the links in the description box below to find out more about what we do in the Obesity and Metabolic Health Lab. So here is where we grab our lab coats, and I'm also showing you the office of our principal investigators, and it's really convenient to have their office right inside of the lab because when I do research I have a lot of questions, so I just come to them and they're really helpful in answering them for me. So we actually share this lab with another professor, Dr. Shin, who looks at the neuroscience of metabolism and obesity, and so I'm about to show you their mice that they have. And so here I am about to show you where we have mice. This is Dr. Shin's mice. Dr. Shin obtained his doctoral degree in neuroscience, and so he's interested in understanding how the brain controls metabolism and nutrient partitioning. One of his projects involves the association between type 2 diabetes and Alzheimer's disease, and this is kind of where my project falls under. My research involves studying the gene expression in the brown adipose tissues of mice with Alzheimer's disease. So it's really cool to see how different principal investigators with their own unique disciplines can kind of come together and ask similar questions, but through different lenses. So one thing that I do in research is I work with cells, and so when we have cells, we need solutions to work with them and to transfer them, and so we keep those solutions in either a fridge or a freezer. And so these solutions are super cold, and cells would get really hurt by that, so what we do is we put them in a little mini bath, as I'm showing you here. This is the lab bench that I share with a PhD candidate, and this is where I do micropipetting. This is also where I do the protein extraction of my brown adipose tissue samples using a needle and a syringe. This is also where we just prep for Western blots or PCR or any other type of experiment that we need to do that day. Here I am showing you one of our 40-degree fridges, and this machine here is called a rocker, and it's used for Western blots. So Western blot is a method that I use to detect the specific protein molecules within my brown adipose tissue sample from the mice with Alzheimer's disease. And so rockers are really useful because when we have a membrane, we need it to be evenly coated with the antibodies to be able to detect the protein that we're looking for. And so one part of the process is where it needs to be incubated overnight. And so this rocker here moves back and forth to allow that antibody solution to evenly coat the entire membrane. So that way we can come back the next day and use another machine to look at the proteins that the antibody found. Now for this next part of the tour, I am going to head you over to our cell culture room. On our way there, this is a microwave, definitely not used for food. And then here we have the flammable closet that contains all of our chemicals that are flammable. And so here I am heading over to the cell culture room where we basically propagate cells and grow them in flasks or wells. And here are our two incubators where we keep the cells that have been transfected with E4-ORF1. That is a viral protein, and we study that protein in the lab. It's from an adenovirus, and this protein improves diabetes, reduces insulin requirement, and reduces fat accumulation in liver. And so transfection is basically used to study the gene expression and protein function by introducing foreign genetic material into a cell. And these incubators are kept at a certain temperature that allows the cells to propagate or grow in a nice and suitable environment.